All right, and welcome everybody again to our third anatomy tutoring session. We don't have a ton of updates for you tonight, and we're just really excited to talk about everything muscles, anatomy, histology, and embryology. So not too many updates. The last thing that I just wanted to say is if you guys wanted to fill out our research survey, that is still open and has been extended for when it's open. That information can be accessed in our news tab on our website, and I can additionally drop that link in the chat as well for anyone interested in participating or wanting to share that link with someone else that's interested in learning more about the scientific process and seeing how that works for a research study. Please just let us know. We'll put that link in the chat again for you guys. So with that, I'll turn things over to Dr. Frank, our moderator.
Hey. Well, good evening, everyone. We are going to start out with anatomy, so gross anatomy, and our first presenter will be Kyle Yu. Kyle is originally from St. Louis, Missouri, but then moved to Seattle, Washington, where he finished high school and graduated from the University of Washington in 2022, majoring in chemistry and biochemistry. He is currently a second-year medical student at Sidney Kimmel Medical College at Thomas Jefferson University, with an interest in anesthesiology or physical medicine and rehabilitation, also known as PM&R. In his free time, Kyle enjoys playing badminton, cooking, or enjoying the outdoors. And with that, the platform is yours, Kyle.
All right. Good evening or afternoon, everyone. Thanks for being here tonight. And can everyone see and hear me okay? All right, sounds good. All right, so my presentation is going to be on the anatomy of the muscular system today. So here are the lecture objectives for today. And I'll start by going over and defining movers, synergists, and antagonists. So the muscles in the body obviously help you move. And so we have different different classifications of muscles depending on how they help your body move. So first you have your primary movers, and those are the muscles that are primarily responsible for a major movement. So in this case, using the example of flexion at the elbow, your brachialis muscle will be your primary mover. You then have your synergists, which assist the movers, and synergists themselves may be primary movers in other functions, but for certain functions, they may be synergists. So in this case, it'd be the biceps brachii muscle, and that would be synergistic to the brachialis muscle to help with flexion. And then lastly, you have antagonists, and those are they work opposite of a mover. So in this case, we have the triceps muscle, which is on the opposite side of the arm as the brachialis and the biceps. And then that is basically antagonistic to the flexion of the elbow, kind of contra the function is opposite of the brachialis and the biceps muscle. All right. Next, we'll go into fascial compartments. And so fascial compartments are going to be groupings of muscles that are separated from each other by connected tissue sheets called fascia. So in this case, I have a cross-section of an arm. And so here, along this dotted line, we have the two compartments, the anterior and the posterior compartment. And then for your upper limb, you have 4 total compartments, an anterior and posterior in the arm, and the anterior and posterior on the forearm. And I think fascial compartments are really important to understand because you can group all the muscles in there, and then you have a more cohesive understanding of what they do and then the blood supply. And so to give the example of the arm, again, the muscles in the arm, muscles in the same compartment, that they will have similar or either the same or very similar functions, as well as very similar blood supplies and nerve supplies. So when you're studying anatomy, because there's going to be a lot of gross anatomy is a very complex, and there's a lot of just facts that need to be facts that are out there. So in allowing yourself to group these muscles into compartments can kind of maybe make the process a little bit easier, and then kind of build an understanding from there. So in the arm, we have your anterior compartments. So this is comprised of the biceps brachii and brachialis muscle, like I mentioned earlier. So that's going to be responsible for elbow flexion, and that will be supplied by the brachial artery and the musculocutaneous nerve. And then the posterior compartment is going to be responsible for elbow extension. And this includes the triceps muscle. And then it's going to be supplied by the deep brachial artery, which is maybe a little bit beyond what you need to know, but it's there for completeness, as well as the radial nerve.
Kyle, it's Dr. Peterson. Before you move on, can you just use your cursor and show them again, because the text is kind of small. Show them the humerus and how that is a good landmark to look for in this cross section through the arm.
Yeah. So the humerus is this, this ring structure right here, and it's kind of the fascial compartment kind of bisects the humerus like right in half. So all of your anterior compartment muscles are like on the anterior surface. They're attaching onto the anterior surface of the humerus, and then all your posterior is attaching to the posterior surface. So again, kind of like going back to the movers and antagonists. If you have like muscle groupings on opposite sides of like a fascial compartment, they will do opposite things. So again, there's kind of like, again, tying back to movers and antagonists, like kind of the understanding the classification there. All right, so we're going to get into some muscles now, and we're going to start with the shoulder. So the first muscle I'm going to go over is the deltoid, and the deltoid is this really huge muscle that kind of goes over the arm. It's kind of like when you see like the shoulder on the arm, your deltoid is covering like that, basically that entire surface. And the deltoid has actually a lot of functions. It's responsible for shoulder abduction, which is bringing the arm away from your body, as well as flexion, which is bringing it forward like this, and then extension, which is bringing it like that, if you can see. And so the deltoid has a lot of functions. It's a really big muscle. And I've included here, and for most muscles in this presentation, the origin and insertions. You're not going to be responsible for these, but I think these are helpful in kind of building an understanding of how these muscles, like the general movement of these muscles. So with the deltoid, it originates in the scapula and the clavicle, and then inserts onto the humerus. And so I guess a way to think about like muscle movement and muscle contraction is I like to think of the origin as kind of like an anchor point for that muscle. And so when you're like contracting the muscle or you're activating the muscle, you're kind of pulling the muscle towards the origin, and then you're also bringing along any attachment more distally to that insertion point along with it. So in this case of shoulder abduction, which is primarily done by this green head of the deltoid, you're contracting the muscle, it's going towards the scapula, and then you're kind of pulling the humerus and then subsequently the rest of the arm up with it in abduction, right? And I'll go over this a few more times with some other examples in terms of contraction and muscle movements. Next, we have the rotator cuff, and this is primarily going to be 4 muscles. So we have the supraspinatus, the infraspinatus, the teres minor, and the subscapularis, which is on the anterior of the scapula. You can't see it on this view. And a mnemonic that I like to use for the rotator cuff muscles is this mnemonic called SIT. So SI little t s for supraspinatus, infraspinatus, teres minor because it's small, and then the subscapularis. And so these muscles are primarily going to be involved in rotation of the shoulder, hence the rotator cuff. And then we also have the teres minor, which is technically not a rotator cuff muscle, but I mentioned it here just because it does have some function within rotation as well.
Yes.
And then, so for the functions of the rotator cuff muscles, the supraspinatus actually has some responsibility in shoulder abduction, just like the deltoid, but actually the supraspinatus kind of kick-starts that movement up to 0 to 15 degrees. And then after that 15 degrees, the deltoid kind of takes over for the rest of the motion. But the supraspinatus has that initial motion. And then we have the infraspinatus and the teres minor, which are responsible for laterally or externally rotating the shoulder, kind of like this. And then the subscapularis and the teres major are responsible for medial, which is kind of the other way, bringing your shoulder in like that. All right, so next we're going to go into the arm muscles. Like I mentioned earlier, we have the biceps brachii muscle and the brachialis muscle. So the biceps attaches at the scapula and inserts onto the radius. And then the brachialis comes from the humerus and inserts on the ulna. And again, this is responsible for elbow flexion. Again, this is the anterior compartment. You kind of bring it towards you. And then the posterior compartment we have, and shown here is 2 out of the 3 heads of the triceps. Remember, tri, the prefix, meaning 3. The medial head is kind of tucked in behind the lateral head here, and it's not shown in this picture. But the triceps muscle comes in from the humerus and from the humerus and the scapula, and it attaches on the ulna. And the function of that is elbow extension, which is kind of bringing your arm back out straight. All right, so next we're going to move on to the forearm. And again, to review terminology, the arm is going from the shoulder joint to the elbow joint, and then the forearm is going to go from the elbow joint all the way down, the rest of your arm. So here is the breakdown of the fascial compartments in the forearm. This is the radius on the left here, and then this is the ulna. And again, the bone is kind of like the big separating point between the two fascial compartments, just like it was for the humerus in the arm. So in the anterior portion, which is what I'm highlighting right here, the function is primarily wrist and finger flexion. And if you can see here, there's actually a bunch of muscles. They're called flexor muscles. So they are very handily named after the function that they do. So in here, and then it's mostly supplied by the ulnar artery, and then supplied by the median nerve mostly. There are small exceptions, but mostly the median nerve. And then the posterior compartment down here is the function is primarily wrist and finger extension. And you can see a lot of these muscles are labeled extensor because of the function that they're doing. And then this is also supplied by the ulnar artery, and then primarily supplied by the radial nerve, mostly. All right, so there's a lot of muscles going on here, but the two I want to point out, one is this flexor digitorum superficialis, which is kind of tucked away in here. So it's kind of below the first layer of muscles in the anterior compartment of the forearm. And so this comes from the humerus and the radius, and this actually inserts all the way into the finger bone. So the muscle comes all the way to about here, and then from there it gives off a bunch of tendons that go to each one of the fingers to the not your thumb, basically. And this is primarily responsible for finger flexion, which is kind of making a fist. That's your main mover function, and it also does help with wrist function as a synergist because it does cross the wrist joint. And the wrist flexion actually is more responsible for these muscles that are like above the flexor digitorum superficialis. And then next we have the extensor digitorum, which is pretty much, it does this, it's the same, it's kind of like the corollary to the flexor digitorum superficialis, except it does it on the reverse. It's on the posterior compartment and is responsible for extending the fingers, again, as the mover, and then also a little bit of wrist extension as a synergist. And here are some examples of the movement, flexing your fingers, and you're flexing your fingers to make a fist, and then flexing your elbow or flexing your wrist like this. Whereas extension is kind of your fingers are more straight out, and then your wrist it's more like that. All right, so this is the first question that I have.
All right, and I'm going to go ahead and close the poll. So get your answers in 5, 4, 3, 2, 1.
All right, so the correct answer is flexion. So remember, the anterior compartment is comprised of your biceps and your brachialis muscle, so it's going to be responsible for flexing at the elbow joint. I also just want to take a quick note of anterior in terms of anatomical positioning. So anatomic positioning is you're standing out straight, and then your palms are facing forward. So that's why like flexion, the anterior compartment of the forearm is also responsible for flexion. So I think that's just something to keep in mind. All right, so next we're going to move on to the lower limb. We're going to start with the gluteal region. So this is kind of like the shoulder region of the lower limb. It kind of connects from the main axial spine to the lower limbs. And so the first muscle I want to bring up is the gluteus maximus, which is the huge muscle back here. It comes from the pelvis and the sacrum, and then it attaches at the femur and then the iliotibial band, which is this long tendinous portion here. And then the primary function here is going to be hip extension, which is kind of kicking your leg all the way back. The gluteus medius muscle is actually just underneath the gluteus maximus muscle. If you think the gluteus medius would just be underneath here. It also comes from the pelvis and then it inserts onto the greater trochanter of the femur. And that's going to be responsible for hip abduction. And then lastly, we have the tensor fasciae lata, which is this small muscle here. And then it attaches from the pelvis all the way down to the IT bands. And the IT band itself goes actually all the way down to the lateral epicondyle of the femur. And then this is mainly for stability of like the lower limb and the gluteal region, as well as hip abduction. And then I just want to also remember that abduction is kind of you're abducting away from the midline. So like oh my gosh, like the aliens are abducting my leg away from me. So that's kind of like the way I remember it. So abduction. And then adduction means that you're adding towards the midline. So like you're adding yourself back together if you're adducting. And so this gentleman over here is showing off his moves and fully abducting his leg up into the air. So just kind of something to remember. All right, so just like in the upper limb, the lower limb has fascial compartments, but this time there are 3 of them in the thigh. And again, nomenclature for the lower limb is that the thigh is going to be from the hip to the knee joint, and then the leg, kind of somewhat hard to wrap around, is actually your knee to your ankle and the rest of the foot. So when I say leg in the later parts of my presentation, I'll be referring to below the knee. So in the thigh, we have 3 compartments. The first is the anterior compartment, which is in red. So this is going to be responsible for hip flexion and knee extension. And then these are going to be both supplied. These are going to be supplied by the femoral artery and femoral nerve. And so something here is that like a lot of different compartments in the body are actually going to be supplied by like the artery and nerve are going to go by the same name. So it's something to keep in mind for, especially when you're studying and trying to understand the compartments and understand the muscles and actions. Sometimes they're going to have the same name, which is going to be very convenient. Next, we have the posterior compartment, which is here in blue, and this is going to be responsible for hip extension and knee flexion. It's going to be supplied by the deep femoral artery and the sciatic nerve. And lastly, we have a third compartment. This time it's the medial compartment, so it's going to be on the inside of the thigh. And this is going to be responsible for hip adduction, and it's supplied by the obturator artery and nerve. All right, so going into the individual muscles of the thigh. So the first muscle that I want to mention is the sartorius muscle, which is this really like small and narrow muscle. It starts at the pelvis and inserts on the tibia. So something that you notice here is this muscle is one of the few muscles in the body that actually crosses 2 different major joints. So it actually crosses the hip joint and the knee joint. So it has function in both. And I guess the best way I can describe the function of the sartorius is if you're trying to sit crisscross applesauce and you're trying to like move your knee into like a bent position, like to crisscross your legs, like you're kind of engaging your sartorius muscle to put your leg in that position. Or if you say you're sitting in a chair and then you're putting one of your the ankle of one of the legs onto your other knee. That's kind of the main function of the sartorius. The next muscle is the rectus femoris, which is this big muscle right here. This is otherwise known as the kicking muscle. This also crosses 2 joints. It starts in the pelvis and attaches into the femur, the patellar tendon, as well as the iliotibial band. And then this is responsible for hip flexion and knee extension, again, because it crosses 2 major joints. So it has 2 different movements here. And then lastly, we have the vastus group. And so the rectus femoris plus these 3 other muscles, all 4 of those make your quadricep muscles. So when you think quadriceps, the prefix of 4, so that's where the 4 in your quadriceps come from. So the 3 vastus group muscles come from the femur, and they insert into the patellar tendon with the rectus femoris muscle. And then the vastus groups themselves are only going to be responsible for knee extension. So seeing this little GIF here, kind of kicking the knee out, that's knee extension. And then this seated position, like this individual would be flexed. So when you're seating, you're naturally in a hip flexed position. All right, moving on to the posterior compartment. So this is otherwise known as the hamstrings. So you have three main hamstring muscles. You have the semimembranosus, the semitendinosus, and the biceps femoris. So even though like there are technically four muscles, if you think about semimembranosus, semitendinosus, and biceps, two muscles, there's only three muscles in the hamstring group because one of the biceps femoris actually doesn't cross the pelvic joint and or the hip joint, and only attaches in the femur. So there's only three muscles here. So with the hamstring group, it comes from the pelvis and the pelvis, and then the femur will be one of the heads of the biceps femoris, and they insert onto the tibia and fibia. And again, because they cross the hip and knee joint, they're responsible for hip extension and knee flexion. So here, this individual is kicking their leg out, so this would be hip extension. And then say this person were to bend their knee and like kick themselves in the butt, that would be knee flexion. All right, the last compartment would be the thigh adductors. So the adductor group is this like, there's three different muscles, but they all kind of come together to form the adductor group. And again, this is responsible for hip adduction. It starts from the pelvis and then attaches on the femur. And again, adduction, you're like adducting back to the main line. So say if your leg is like all the way kicked out, like that one gentleman was in the earlier slide, in order to bring your leg back together, you would contract on your adductor group, and then that would pull closer and closer together. And we'll get into contraction when we get to, I believe, Zach's presentation next in histology. But yeah, you know, when you're contracting the muscle, you're activating the muscle, and you're able to pull the femur and subsequently your leg back towards your midline with the rest of your body. All right. So the last compartment, or last region we're going to talk about is the leg. So again, the leg is going to be anything below the knee. And then we have three main compartments in the leg as well. So first we have the anterior compartment, which is in red over here. This is going to be responsible for dorsiflexion and toe extension. So this on the bottom left, we have a diagram. So dorsiflexion is going to be like kind of angling your foot upwards at the ankle. And this is going to be supplied by the anterior tibial artery and the deep fibular nerve. So, you know, I mentioned in the previous, in the thigh with the anterior compartment that there's a lot of similarities, but this is like one of the main naming differences between like the artery and the nerve, because you have the anterior tibial artery and the deep fibular nerve. So I just wanted to make sure that this is like one of the big exceptions to that rule, and to with the branching pattern in the leg kind of gets a little bit wonky down here. Next, we have the posterior compartment, and this is going to be, this is in purple, and this is going to be responsible for plantar flexion as well as toe flexion. So plantar flexion is kind of angling your foot downward, and then toe flexion is like thinking of like, oh, if you're trying to pick up something with your toes, you'd be flexing your toes to try and grip that object. And this is going to be supplied by the posterior tibial artery and the tibial nerve. And then lastly, we have the small lateral compartment. So lateral being on more outside. So this would be, it would have a synergistic effect on plantar flexion, but its primary effect would be eversion. So eversion is kind of pulling your toe to like more the inside. So you're putting more weight onto your big toe. And then inversion is kind of if you're standing on the sides, or you're walking on your sides of your feet, you'd be your foot would be in an inverted position. And then this is supplied by the fibular artery as well as the superficial fibular nerve. And I guess one more thing to note, this image has a distinction between the deep and superficial posterior compartments. And for the intent of this presentation, I've just kind of combined those as one. All right. So the muscles of the leg/calf, they're very similar to the forearm in the fact that we have an extensor digitorum longus muscle. So this comes from the tibia and fibula, and this inserts on the foot bone. And then like the name says, it's going to be responsible mainly for the extension of the toes, as well as a little bit of synergistic effect on dorsiflexion. Likewise, we have the flexor digitorum longus, and then this is going to be primarily for flexing the toes, as well as synergistic effect on plantar flexion. And then the last muscle, so the muscle we think of as the calf muscle, that's called the gastrocnemius. And this comes from the femur and attaches onto the Achilles tendon. And then this is the primary function of this will be plantar flexion. Actually, it's one of the few muscles in the body that when you're standing, it's already naturally contracted because it's kind of a structural supportive postural muscle, and it kind of keeps your legs nice and sturdy. All right, another question.
All right, you guys are doing a good job with this. I'm going to go ahead and close the poll in 5, 4, 3, 2, 1.
All right, so the gastrocnemius would be in the posterior leg. Again, remember the thigh is going to be the region between the hip and the knee joint, and then the leg is going to be from the knee joint and down below. So it would be in the posterior leg compartment. All right, so for the last learning objective, we're going to go into nerves and arteries. So first, the nerves of the arm. So in orange, we have the musculocutaneous nerve that goes down here. And then on the anatomical diagram, it comes from here right here. The biceps has been removed, so it kind of goes right behind the biceps muscle. And so this will supply the anterior compartment of the arm. The radial nerve, which is in yellow, is going to supply both the posterior compartment of the arm and the forearm. And then this will, you can't see it in this photo, but it kind of, because it's in the posterior view, it's going to go kind of behind. And then the median and ulnar nerves, so median in green and ulnar in blue, they're going to supply the anterior compartment of the forearm, and they're also going to have some contributions into the hand as well. So the ulnar nerve is right, or the median nerve goes right here, and then the ulnar goes like there. All right, so in the middle region, so we have the femoral nerve. So the femoral nerve is going to come from like the lumbar area and then go through inguinal canal and into the anterior thigh compartment. And as well, we have the obturator nerve, which kind of comes in, and then it kind of hugs along the medial compartment of the leg to supply the thigh adductor / the medial compartment. And lastly, we have the nerves of the lower leg and the posterior side. So first we have the sciatic nerve. And then, so this is the largest nerve in the body, actually. And then this supplies mainly the posterior thigh muscles, except for one muscle. And then the sciatic nerve actually divides in two. So it divides into the tibial nerve. And so this will keep going down and then supply the posterior compartment of the leg. And then it will also branch into the common peroneal or fibular. So you may see some, depending on what text you use, peroneal and fibular are like the same thing. So it just kind of, again, depends on what source you're looking at. And so the common fibular will then divide into the deep and superficial fibular nerves, and then they'll supply the anterior and lateral compartment of the leg respectively. You can't really see it on here, so I just kind of put the arrows at an estimation because it'll kind of wrap around to the front and then branch off into the deep and superficial branches, but around there-ish they'll branch off. All right. Do we have time for another question or?
Yeah, I think we could do one more. Practice makes perfect. All right, and for the sake of time, I'm going to go ahead and close this poll in 5, 4, 3, 2, 1.
All right, so yes, it would be the femoral nerve. So again, I want to reiterate that a lot of the nerves also travel with like similar arteries. So femoral nerve travels with femoral artery. The obturator comes with the obturator artery. So they'll kind of be in pairs. And if not, then there's going to be some correlation depending on the compartment. So kind of thinking about the compartments, what muscles are there, and then how like what supplied nerves in the artery. All right. So lastly, I'm going to talk about the arteries. So this diagram has like a whole list of different arteries in the body, but I just want to focus mainly on the ones that are in the red boxes. So we have the heart, and so the heart pumps out into the aorta, and the aorta kind of branches off into the brachiocephalic trunk on the right side, which then branches up to the carotids, which go up to your head and neck, and then subclavian, axillary, and then the brachial artery. And the brachial artery splits into the radial and ulnar artery to supply the upper limb. And then when you go down, you have the descending aorta. It goes through the abdomen and then branches into your common iliac. And then your common iliac branches into your external and internal iliac. With your internal iliac provides a bunch of different blood supplies into your pelvic region, but the main one to really get away is the obturator, as that supplies the medial compartment of the thigh. And then the external iliac becomes the femoral, and then the femoral changes name into the popliteal when you get to the popliteal, or like behind the knee region. And then the popliteal becomes splits into the posterior and anterior tibial arteries, and then the posterior tibial splits again into your fibular artery. And so this is kind of like a very simplified diagram that kind of gets away with most of the other arteries. And this is maybe a very useful study tool for you in saying, okay, this is like the general way of like the blood supply, especially with the aorta. There's like a little bit of asymmetry because of the heart. And it's like you have to go through the brachiocephalic trunk on the right side versus going to just the common carotid and subclavian on the left. And again, the brachial artery will supply the arm, and then the ulnar and radial will supply the forearm and the hand. And then in the abdomen and into the lower limb, so you have the descending aorta, again, comes the common iliac, and you go into your obturator and thigh adductor groups, and then the external iliac, the femoral will supply the thigh, and then you have your compartments of the lower limb as your like terminal branches of the arteries. All right, I do have one more question, but I don't know if there's time, but.
I'm going to see if people could start putting questions in the chat or raising your hand. And then while you're thinking of those questions, I'll go ahead and just put one more poll up for this one. Because again, I think the asking questions is just such a good way to practice the material. So we'll do this one a little bit quicker within about 30 seconds if you guys could get your answers in. Alrighty, and I'll close the poll in 5, 4, 3, 2, 1.
All right, so yeah, so it would be the posterior tibial artery. And again, the branching pattern in the lower leg, it's a little bit more complicated. But yes, the posterior tibial one's the tibial artery. Remember, the anterior tibial actually lines up with the fibular nerve. So that's again something to keep an eye out for. And yeah, femoral comes with femoral, and popliteal is just behind the knee. That's in my presentation. Again, I just want to make one more point of, trying to understand the compartments and trying to figure out like the blood supply. I think that was like, there's definitely a lot of information in this presentation, but I think trying to literally compartmentalize all this information into the respective compartments and like, the artery and nerve, I think will hopefully really help in your studying. Yes, thank you for listening.
We do have a few minutes for questions if anybody has any. For Kyle?
You can also ask them in the chat if you don't want to unmute yourself.
Yeah. So we have a question. Could you go over the difference between a vein and an artery again?
Mhm. Yeah, so I didn't cover any veins in my presentation, just because I know like on the structure list, it kind of more covers the artery and with the learning objectives, but arteries are going away from the heart, whereas veins are going towards the heart. So veins, when you pump out blood from your heart, your arteries will take that like fresh oxygenated blood from your heart and then distribute it out to the body, whereas the veins will take that like deoxygenated, used up blood like from the periphery of the body back to the heart to get reoxygenated.
Okay, well, if there are no further questions, then we can take Kyle off the hot seat. Excellent presentation this evening, Kyle. Excellent, excellent work. Thank you.
Thank you.
Then we can move on to our second presenter, who will be covering histology. And is he on here?
Yeah, I'm here.
Oh, there we go. Okay, so our next presenter is Zach Mattiola. Zach is from Collegeville, PA. He graduated from the University of Michigan in 2022 with a degree in neuroscience, and he is currently a second-year medical student at Drexel University. In his free time, he likes to watch sports, especially Michigan football, the Eagles, and the Sixers. He also likes to work out and play basketball and pickleball. Academically, he is interested in pharmacology, anatomy, and physiology, and he wants to specialize in anesthesiology, critical care medicine, or surgery. And with that, Zach, the floor is yours.
All right. Thank you, Dr. Frank. Can you see my screen? Okay. All right. All right. Thank you guys for spending your Sunday nights with us. Means a lot to us. So I'm going to be talking about muscle histology, keeping in the trend of the muscle tonight. So here are our lecture objectives. I'm going to be going over the three types of muscle in our body, skeletal, cardiac, and smooth muscle. Look at what they look like basically under a microscope, what their function is, and where you can find them in the body. And then I'm also going to be going into depth a little bit more about skeletal muscle as well. All right. So here's just a nice little summary table. I'm going to go over this at the end with you guys as well, just to kind of give you a brief introduction to these types of muscle cells. So I think it's important to understand what the nuclei look like, what their shape is. You're going to soon learn about the striations of the muscle cells and how the body controls their contraction. Okay. So we're going to start out with skeletal muscle. Okay, so this is what the skeletal muscle cell looks like under the microscope. This is one skeletal muscle cell right here. This is another one right here. Okay, so these skeletal muscle cells, they're called myofibers or muscle fibers, and they're basically one long tube or syncytium. It's like the way I like to think of it is if you take a bunch of individual cells and you combine them all into like one cytoplasm, that's basically what a muscle cell is like. It's basically one continuous hollow tube with a bunch of nuclei that are located on the periphery of the cell. You see here, these are the nuclei. And they are basically just, like I said, one long tube. And they are filled with this contractile protein called myofibrils. Okay, they're just chock full of these myofibrils because the function of these cells are to contract. Okay, so they're one long empty tube filled with these myofibrils, and these myofibrils will give them this characteristic striated appearance under the microscope. Okay, and these myofibrils are made up of these contractile elements called sarcomeres that I will explain to you about a little bit later, okay? So what is the function of skeletal muscle? Skeletal muscle is under voluntary control. We can decide to move our arms and legs, and skeletal muscle allows us to move, right? Skeletal muscle is located all over the body. Like Kyle said, they attach to bones, and their contraction allows us to move our limbs, to move our body, all that good stuff. Okay, and their naming is based on their shape, their size, how many like bellies that they have. So you've heard of the biceps, the triceps, the quadriceps. The biceps is named because it has 2 bellies. The triceps has 3 bellies, and the quadriceps has 4 bellies. So it makes it a little easier. And muscle cells muscles themselves must cross at least one joint in order to function properly. Because if you think about the biceps, it starts in our shoulder and it attaches in our forearm. And by attaching in our forearm and crossing the elbow joint, its contraction allows you to flex your arm and pull your forearm towards you. Okay. So we have our first question here. So which of the following is the most accurate description of the skeletal muscle fiber, the muscle cell?
All right, you guys are doing pretty good with participation. We're gonna go ahead and close the poll in 5, 4, 3, 2, 1.
All right. So the right answer, for those of you who said B, you are correct. And I know it is a little confusing. So normally we think of cells as having only one nuclei, right? But muscle fibers are a syncytium. So it's basically like a bunch of cells come together, they combine, they fuse all their cytoplasms together, and they comprise one cell. Okay, so one muscle fiber has one big long cytoplasm, and there are a bunch of nuclei in there that are sitting out here on the edges. Okay, but you guys did really well. Answer C also says there are striations and they're long and unbranching, and that's very true and very important to keep in mind. But yeah, so we have multiple peripherally located nuclei. We have these striations from those myofibrils, and they are basically just long unbranching tubes. Okay, so now a little bit into the organization of the skeletal muscle. I talked to you a little bit about the muscle cell or the muscle fiber. Okay, so that's basically the fundamental unit of the muscle organization. So when you get a muscle, excuse me, when you get a bunch of muscle fibers that come together, they're going to form what's called a fascicle. So here's one muscle cell, and he's joined together by a bunch of other muscle cells, and they form a fascicle. And then you get a bunch of fascicles together, and those comprise the actual skeletal muscle. Okay, so we go from muscle fiber to fascicle to the actual skeletal muscle. Okay, and all of these organizational layers are going to be surrounded by connective tissue. So if we start at the largest organizational layer, the actual muscle, that's going to be surrounded by a layer of connective tissue called the epimysium. Okay, and then each individual fascicle is going to be surrounded by perimysium. And then the individual muscle cells that make up the fascicles are going to be surrounded by epimysium. Okay, and it's important to keep in mind that this connective tissue is going to be a fibrous connective tissue, so it's really tough and gives the muscle cells good support. for what they need to do. All right. All right. So now we're coming back to those contractile elements inside the muscle cells that I hinted at a little bit earlier. So if you remember, I know I'm beating a dead horse here, but I think it's important. A muscle fiber, a muscle cell is just a long tube, and it's just full of these contractile proteins called myofibrils, okay? The muscle cell, all it needs to do is contract. So it can just jam pack its cytoplasm full of these contractile proteins, okay? So these myofibrils are also long tubes, but they're composed of these fundamental units called a sarcomere. And these myofibrils are basically just one sarcomere lined up after another, okay? And these myofibrils, the way they're arranged, give the muscle cell these nice striations that you see under a microscope. So what is the sarcomere? Okay, the sarcomere is basically overlapping protein filaments, okay? We have two major types of filaments called a thin filament, also known as actin, if you've heard of that before, and a thick filament, also known as myosin, okay? So with them overlapping, that allows the myosin to grab on to the thin filament when it's time to contract and basically pull it close together. So the myosin is going to grab onto the thin filament on this side and grab onto the thin filament on this side and pull them both close together. And this is going to contract all of the myofibrils inside of the muscle cell, contract the muscle cell, and ultimately contract the muscle. Just this interaction right here. And that's the basis for muscle contraction inside of your skeletal muscles. Okay? So there are a couple of important zones relating to the sarcomere that we think about that kind of helps us visualize the contraction and understand it a little better. The first zone is called the A band, and the A band is going to correspond to the full length of the myosin thick filament. So this entire filament right here is going to be the A band, okay? The I band is going to correspond to parts of the thin filament that do not overlap with the thick filament. So if you see this little portion right here of the thin filament is overlapping a little bit with the thick filament, that's not a part of the I band. The I band starts right here where this actin isn't overlapping with the thick filament. And that's going to comprise this entire portion right here until we get to the portion that's not overlapping with the thick filament on the other side. Similarly, the H band corresponds with the portion of the thick filament that does not overlap with the portion of the thin filament. So just like with the I band, the H band is going to start about here and go to about there, okay? And think about it like this. When this guy contracts, when this myosin is pulling this actin closer to the center here, this H band is going to get smaller, right? And we can see this under a microscope when you're looking at a muscle contracting. That's kind of, I think, how these zones were first like thought of. And similarly with the I band, it's going to get smaller as more overlap occurs between the thick and the thin filaments, okay? And because these are proteins, because these are filaments, they need to attach somewhere or else they just float away. So the thin filament is going to attach at what is known as the Z line. That's this point right here, or in this schematic down here, okay? This is where these thin filaments are basically gonna be anchored to, okay? And the M line, which is right here in this schematic, or right here, or right here, is where the thick filaments are going to attach, okay? So that's sarcomeres. Now I just want to, and here's a really good non-cartoon, I think this is an electron micrograph of the sarcomere. So you can see the M line, the Z disc, the I band with the thin filaments, the A band with the thick filaments. So now we're going to look at a video of a sarcomere contracting, and I think this is really great to understand how this whole process works. So here's our sarcomere, here's our actin, our thin filament, and in the middle we have our thick filament, our myosin. So let's look what happens. So you can see the thick filament is attaching to that actin, and it's pulling it closer together, and it's contracting it. And that's happening when you flex a muscle. That's happening throughout the entire muscle and all the muscle cells. All of those little fibers inside of those muscle cells are contracting and being pulled closer together. Okay. All right. Another question here for you. So let's see if you remember those connective tissue coverings. So which layer of the fibrous connective tissue covers the individual skeletal muscle cells? I know this is probably a little bit harder. It's just rote memorization.
Looks like some of you guys have figured out your prefixes here. And so I'm going to go ahead and close the poll, get your answers in. I'm going to close it in 5, 4, 3, 2, 1.
Oh, so good. It is the endomysium. Remember, endo is like inside, basically, right? It's going to be the innermost layer whenever that's the prefix, and that is the case. That is the case here with the endomysium. It is going to be surrounding the muscle fibers, and then the perimysium are the fascicles here that are comprised of those muscle fibers, and the epimysium is going to be the entire muscle itself. Okay. All right, so now that we've discussed skeletal muscle in depth, where's my cursor? Okay, let's discuss cardiac muscle. Okay, so cardiac muscle has a lot of similarities to skeletal muscle. It also has a lot of differences. One of the differences is that unlike skeletal muscle, remember what I said, it's just a long tube full of cytoplasm, nuclei, and contractile elements. Cardiac muscle are individual cells that are connected by these little points of attachment called intercalated discs. Don't worry about what the intercalated discs are. Just know that this is an individual cell here. This is an individual cell here. This is an individual cell here. Okay. They also are branching. You can't really see the branching that well in this image, but trust me, they're branching. And they have centrally located nuclei. So remember, the skeletal muscle cells had peripherally located nuclei, but these guys have centrally located nuclei, okay? And they also have striations, which means they also have sarcomeres. Remember, the striations mean that they have sarcomeres, okay? And here's a nice little cross-sectional image of cardiac muscle. You can see the centrally located nuclei here, and maybe that's a branch point. I don't know, maybe, but that's cardiac muscle. So I'm sure you all quickly figured out that cardiac muscle is found in your heart, okay? And if you've ever tried to stop your heartbeat voluntarily, you know that it is not possible because your heart is under involuntary control. So your skeletal muscle, you can choose to move them if you want, but you cannot choose to contract your heart. That happens automatically. So it is under involuntary control. And its function, the heart's function, it pumps blood all throughout your body to deliver oxygen and nutrients and all that good stuff that your tissues need. Okay, and here's a good picture of the branching that you can see. These cells are branched and kind of a lot less organized than skeletal muscle is. And you can't really see the striations too well here, but in this cartoon picture, there's some striations for you. Okay. All right. So our last type of muscle is going to be smooth muscle. And smooth muscle is kind of like, it's the weird one of the group, okay? It's really so much different than skeletal and cardiac muscle. So smooth muscle, the cells themselves are spindle shaped. So you can see this kind of looks like a spindle. Their nucleus is centered in the cell, just like cardiac muscle, and it kind of looks like a cigar. If you can see, it kind of looks like a cigar almost. And they're also spiraled as well, as if you can see, they kind of look like twisted a little bit. Okay, they have actin and myosin, thin filaments and thick filaments, because they're muscle cells. They need to contract, but their actin and myosin aren't arranged in sarcomeres, so they don't have any striations. Okay. And smooth muscle is found all throughout the body. It's in your GI tract, it's in your urinary tract, it's in your blood vessel, it's in your eyes, it's everywhere. Okay. And its function is different based on where it is in the body. So I wouldn't get too bogged down at this point about the function of smooth muscle, just kind of know what it looks like under a microscope. You know, spindle shaped cells, central cigar shaped nucleus, and no striations. Okay. Yeah, so that is smooth muscle. Here's a cross section of it. It kind of looks it's kind of harder to tell on the cross-sectional image the difference between muscle in the heart and skeletal muscle and smooth muscle, because you can't really see striations. But the smooth muscle is, I think at least, it looks a lot more tightly packed together. And you can see like more tightly packed nuclei in there. And I say that because our next question is trying to differentiate the three muscle types, the cross-sectional image. So go ahead and try and see if you can do that. I understand this is probably a little harder because you can't really see any striations or branching, or you kind of only get to see where the nucleus is, but give it a shot. Hopefully you can identify at least what 3 is, because there's one big feature that kind of gives it away.
Right, and get your answers in. We'll go ahead and close the poll in 5, 4, 3, 2, 1.
All right. Yeah, this is kind of what I expected. C and D to be the main answer choices. So I'm glad you all recognize that this is skeletal muscle, right? It is peripherally located nuclei. Okay. 1 and 2. So 1 is going to be cardiac muscle. 2 is smooth muscle. And I was right before this started, I was looking at this question. I was thinking, this is kind of tough. But how I thought of it was, smooth muscle cells are typically like jam-packed together, whereas, cardiac muscle, you have a lot of spaces in between them, you have a lot of branching points, so there's going to be some more space between them. Whereas smooth muscle, these guys are all just kind of packed in there real tightly together. And then, skeletal muscle, this, you can't really mistake this for anything else. This looks characteristic of skeletal muscle. But yeah, that's a tough question. All right, so back to this table. I hope this makes a lot more sense to you now. So remember, skeletal muscle, multiple peripherally located nuclei, a long hollow tube full of contractile elements with striations under voluntary control. Smooth muscle, you have just these single centrally located nuclei. They look like spindles. There's no striations because they don't have sarcomeres, and they're also under involuntary control. Cardiac muscle, centrally placed nuclei with striations, with branch points. There's single cells connected by these connected at these intercalated discs. And it's also under involuntary control. Okay. I think as just since this didn't take that long. In terms of studying, for this or any other science class you have in the future, I think making tables like this is a really, really good way to test yourself. Just make categories like, skeletal muscle, smooth muscle, cardiac muscle, a bunch of like histological characters or histological aspects of it, and just write in, see if you can fill in the entire table. And that's a really good way to test yourself and to study, in any science course that you take in your life. All right. All right. So that's all I have. So whatever questions you may have, I can answer them.
Zach, it's Dr. Peterson. Why don't you go back to that cross-sectional slide, that question slide, and just explain to everybody that's watching or will be watching on the recording why some of those cross-section cells don't have nuclei. I think that would be really helpful if they understood that. And you've got a little bit of time.
I mean, well, you have a sheet of cells basically, right? And if you cut through them, you're not always going to hit a nucleus, right? Is that the answer? Okay. Yeah. That's, if you cut through a cell, you can cut through the edge of the cell, you can cut through right next to the nucleus, you can cut through right through the nucleus. And we just happen to get lucky with this guy, this guy, this guy, all these guys, we hit the nucleus. Whereas with some of these guys, the nucleus might be behind us, it might be in front of us. It depends. And the same is true for, these guys, you just happen to not get a nucleus in this cross section here. But in some of the others, you were lucky and you did.
That was great. I think you have a question in the chat
Yes.
now too.
So what's the difference between actin and myosin? That's a good question. So actin and myosin, let me go back to, where's my sarcomere? Okay, so actin and myosin are basically two different types of proteins. Okay, just two different types of proteins. Myosin is this big, bulky protein with this head group, and myosin is basically just like a chain of proteins. And the myosin has this big, bulky head group, and it's going to grab onto the actin and pull the actin close together. And that's really it. They're just two different types of proteins that are responsible for contractions in your cells. And if you know what micro or what's the microfilaments or whatever that filament is, there's a cytoskeleton protein that's made of actin. So that's all it is. They're just two different types of proteins.
Do we have any more questions for Zach?
So do you mean like as in an organization to the contractile elements? Because in cardiac muscle has sarcomeres. So it is organized pretty similarly to skeletal muscle. It has those sarcomeres, it has, the myosin and actin organized in that way. But smooth muscle, it's a lot different, and it's a little more complicated how the myosin and the actin end up interacting with each other. But the cardiac muscles do have sarcomeres. Does that answer your question? Oh, with epi, peri, and endo. No. So, no. So the the heart muscle or the myocardium is just is just organized with these muscle fibers. Basically, it's not organized into different layers, I think, right?
Yes, and that was a great question, Jawan. And maybe Zach can move his, excuse me, move his cursor, and all of those white spaces in between the cardiac muscle cells, there is some fibrous connective tissue there, and that's important because that's where the blood supply and the nerve supply is going to permeate or move through and between those cardiac muscle cells that are woven in that net-like mesh. So they all get a good blood supply, and under involuntary control, we still need involuntary nerves that can increase the rate of the heart and then slow the heart rate back down to a baseline rate. So I thought that was a really good question.
I could add just one more thing too. There is a little bit of connective tissue around these cells. So if you look over on the top right histology image that he has, you can see it's kind of pink, and then there's little hints of little lines of green that are running through. And so the dye, the specific stain used on this tissue, that green is going to highlight connective tissue. So you can see there's little bitty bands of connective tissue that's running in between these cells in cardiac muscle and in smooth muscle too, but it's just a little bit. It's not these like nice, thick, defined sheets that we see in skeletal muscle.
We have another question. Are there T-tubules in cardiac muscle?
Yeah, so that's a great so I think yes, so I guess I don't think you can see them here, but yes, there are T-tubules in cardiac muscle because you're full of you're chock full of those proteins, right? You're gonna have a hard time conducting electric charge deep into that cell. So you need a way to bring that charge deep into the cell where it might have a harder time reaching. So there are T-tubules, just like there are in skeletal muscle. They're just a little different. I don't think you can see one here.
Excellent questions. And do we have any more for Zach before we let him go? Okay. Thank you so much, Zach. Excellent presentation. Thank you for joining us tonight. Okay, so with that, Zach, you're off the hook, and I will introduce our last presenter, who will be discussing the embryology. And
that's me.
Yes, Austin. Okay, there you are. Okay, so Austin Boehm is going to be presenting the embryology of the musculoskeletal system. So Austin is from Millbrook, Alabama, and graduated from Auburn University at Montgomery, majoring in biology pre-health. He is currently a second year at Edward Valle College of Osteopathic Medicine in Auburn. Outside of school, Austin enjoys gymnastics, parkour, which I had to look this up, which is a sport of traversing environmental obstacles by running, climbing, or leaping rapidly and efficiently. Is that correct, Austin?
That's a pretty good description, honestly.
Okay, thank you. And all types of related acrobatic activities. Austin's favorite part of anatomy is being able to apply it to physiology to make sense of how the body functions. And so with that, I give the platform to you, Austin.
All right. And can everyone see the slides? All right. So hello, everybody. Again, my name is Austin. Thank you for sticking with me here. And today I'm going to be talking to you guys about the embryology of the musculoskeletal system, essentially how it kind of came about and originates and develops from a baby. So we don't have too many lecture objectives. Essentially, we just have to describe how the limbs are formed from different tissue layers, and then another piece of those called somites and what their function is with some different structures. But to begin with, I'm just gonna ask you guys a matching question here. If you want, you can just type these out in the chat as you think, or try to think about the different relationships, because there's a lot of different vocabulary in embryology. So I want to kind of try to gauge where you guys are at with your understanding. So just thinking about some of these words here are going to be helpful as we get going. And we have the answers there. So a couple of these words, cranial and caudal, are mainly referring to the head and then the sort of tail end of a developing embryo. For some reason, with adults, we use superior and inferior when we're describing things, but for the developing embryo, the people that made embryology just decided they wanted to come up with their own type of lingo and make up words. So there's a whole bunch of different vocabulary to learn. These other words, sagittal, coronal, and transverse, are referring to different planes, some of which you might have seen in some of the other presentations, such as those transverse sections or the longitudinal sections of the muscle. But essentially, sagittal just means we're cutting something down the midline into left and right pieces. Coronal means that we're cutting it into front and back pieces, and then transverse means we're cutting it into top and bottom pieces. So down here in this picture, green would be transverse, sagittal would be red here, and then coronal would be that blue. So I don't know how much you guys know about embryology, but the embryo and all of us kind of developed starting out as this flat little disc sheet. And from that little sheet, we're going to begin different types of folding. There's two main different types of folding. One of them is cranial caudal folding, which is where we're folding from head to toe, which is right here. And then the other type is lateral folding, which is shown here. And that is kind of how we end up with this hollow section in the middle and how we end up with a whole body cavity, essentially. Now, while we're folding, there are 3 different layers of tissue that are developing from this disc that are going to become really important for structures that are going to generate later on down the line. And those 3 layers are ectoderm, mesoderm, and endoderm. For the musculoskeletal system, the most important one is going to be mesoderm. And essentially all these words are just ectos like outside, mesos in the middle, and then endo is meaning kind of on the inside. So when we're looking at this tissue over here, I have a star by this somatic mesoderm. Mesoderm kind of splits off. There's a whole bunch of different branches and subcategories of the tissue, but the main one that we're going to be focusing on is somatic mesoderm, and somatic refers to kind of the body wall. The other kind that's not as important for us today is called splanchnic mesoderm, and splanchnic is referring to more visceral. and that's going to be related to this gut tube here in the middle. And that's more with the digestive system. So here again is a couple of the different things for endoderm, mesoderm, and ectoderm. But for us today, the main one we're focused on is the mesoderm, specifically the musculoskeletal system, although it does also have some functions with circulation and connective tissue, and as well as a little bit in the lungs. And then here's another kind of sagittal section of a developing embryo. You can see it's growing. This is going to be kind of in the uterine walls, and there's blood supply growing, and this yolk sac in the middle lined with endoderm that's going to become the internal gut tube that we've got. More on the outside here, we've got the ectoderm, which is going to become more neurovascular, the brain and skin. And then the red tissue, which is what we're mainly focused on today, that's the mesoderm, which is going to end up becoming musculoskeletal system. So as for some of those divisions of the mesoderm I was talking about, some of the main ones that we're going to focus on today are paraaxial and lateral plate mesoderm. This is a transverse section of the developing embryo. So if you think of it, we've cut the body and we're looking at it kind of from the head down through the middle. And we've got this yellow section here, the gut, that's the endoderm. And then two of the main important layers of mesoderm is paraaxial and then the lateral plate mesoderm. And that's going to be important for future development. And those things are happening alongside a whole bunch of other development. And some of the landmarks are the neural tube here, which is eventually going to become our spinal cord, and then the notochord, which is just an embryologic structure that's kind of there to differentiate where the midline is and help with other hormones and things like that. So this is happening around weeks 4 to 6, roughly. All these different tissues are beginning to differentiate. There's a whole bunch of things going on. But the paraaxial mesoderm, para kind of means beside, and axial is like, I think of it as the central axis of the body. And the paraaxial mesoderm is forming into a structure called somites. And that's starting from a cranial to a caudal end, meaning it starts at the head and it slowly works its way down. And that's happening beside this notochord, which is kind of an important structure to differentiate the midline for development in the embryo. Now, what these somites are going to do is, as time goes on, they're going to further differentiate into a few different types of layers of tissue that give rise to other structures. So as they differentiate, there's a layer called the dermatome, which is going to form the dermis and subcutaneous tissues, which is just under the epidermis layer of the skin. So these are kind of superficial coverings that are going to be going over our body wall and our arms and limbs and those types of things. The other one here is the myotomes, and that's going to end up forming the muscles of the body. So the myotomes here, they'll also contribute to the limbs and the arms and legs and those types of things, but they also form a little bit of the muscles kind of on the back, some of those superficial muscles. And then the last thing that somites do is the sclerotomes, and that is the bony structures of your kind of central column, your spinal vertebrae and your ribs and those types of things that kind of protect all the important things in that central axis. And those are kind of going to stay there next to the neural tube. They're not going to be branching out too far. Now, as for the lateral plate mesoderm here, that's going to give rise to the skeleton for the appendicular skeleton, the bones for the appendicular skeleton. And that happens around week 6 with the initial development going through with hyaline cartilage. So as most bones form in the body, they start out as cartilage. So it's kind of like a template that becomes formed. And then after that template is filled out and starting to develop, then the osteoblasts and osteoclasts get to work, bone cells, and they kind of start putting bone and filling out that template. And around week 6 is where we start to get limb buds. So you can see a little bit of those in the picture here. And they form off of this portion of the ectoderm that starts to kind of thicken and form a little lump or mound. And that's called the apical ectodermal ridge. I always thought of that in association to the appendicular skeleton, which means like our arms and legs because it's AP for appendicular. And then it's ectoderm because it's sitting on the surface, and anything on the surface is pretty much always going to be ectoderm. Now, as the limb buds develop, it's a bit complicated, which is a bit outside of the scope of what we need to know, but it starts proximal to distal, which essentially just means it starts close to the body, and then it slowly grows its way out. And as those cells on the end start to work their way out, they're going to get further from that apical ectodermal ridge, and it's going to slow down the dividing of the cells, which I think is essentially due to they're getting less of those hormones that are central and important to the development of things. Initially, the limbs are just essentially flat. There's no structure in particular to them, besides maybe a flexion point for the elbow or the knees. But later on down the line, there's apoptosis that occurs and allows for the development of fingers and toes. So when we were all in the womb, originally we had essentially little mittens, and then our fingers formed out of that. Now, as for what these layers actually do and what they become, the lateral plate mesoderm is going to become the bony structures for our arms and our legs. So the femur, the tibia, the humerus, radius, and ulna, all of those bones are coming from the lateral plate mesoderm, which can be a little bit confusing because it is called lateral plate mesoderm, even though it is kind of anterior looking on this. But if you think about earlier, kind of back at the start, how we had that lateral folding of the embryo, that's the reason why this is called the lateral plate mesoderm. It used to be out on the sides, but when we had that lateral folding, that brought them together, and now it's kind of anterior or towards the front, or the ventral side. The other two parts of the arm or leg would be the myotome and the dermatome. So the myotome comes in here from that somite and is going to form the muscles. So those anterior and posterior compartments that were just talked about are coming from the myotome there. And then the dermatome is forming the dermis and subcutaneous tissue, which is kind of giving that cushioning to the neurovascular structures, the arteries, the veins, and the nerves, and helping to get a little bit of padding in between the skin and surface as well. All right, so I have another question here. This one is multiple choice. So which of the following layers of the embryo become the appendicular skeleton?
All right. I know embryology is hard compared to, I think, a lot of the other topics, especially when you're first getting out, but looks like you guys are doing a good job taking a shot at it. I'm going to close the poll in 5, 4, 3, 2, 1.
All right, so it looks like most of you guys were on the right track with that, trying to choose between the lateral plate mesoderm and then the regular mesoderm. But the correct answer is the lateral plate mesoderm. That's because the question is asking specifically about the appendicular skeleton. If it just said which of the following layers will become the skeleton, that would be more appropriate for mesoderm. The lateral plate mesoderm is a subcategory of that, and that's why that's the best answer for appendicular skeleton. Some of the other ones, ectoderm is surface, so that's skin and nerves. Endoderm is that deep inside, which is the gut tube. And then paraaxial mesoderm has axial in the name, and so that can give you a hint that that's going to become more of the axial skeleton. And I've got one more question here. When do the limb buds begin to form during embryologic development? And I've got a few ranges here because for almost anything in embryology, it's kind of a window of when things are most likely to happen.
All right, and I'm going to close the poll in 5, 4, 3, 2, 1.
All right. It looks like most of you guys were on the right track there thinking B. So weeks 4 to 6 is like the time when you would see that the limb buds would start to develop. 2 and 3 is a bit early. That's mainly when you're just starting to get those differentiation between the different layers of tissue. And then any time past like 8 weeks, a lot of the super important types of development are already complete. So something significant about the period before 8 weeks is that's when the embryos are most sensitive to teratogens, which can cause mutations in a developing embryo. All right. And lastly
we have
here,
a question here.
yes.
Are limb buds part of the ectoderm?
The limb buds themselves. So the limb bud, it has multiple layers to it, kind of just like anywhere else in the body. So right here, it does have an ectoderm on it, and the ectoderm in this graphic here is represented by this blue kind of outline. And the ectoderm right here, that's forming your epidermis, so the superficial layer of the skin. Anywhere that you can see skin on yourself, that's going to have come from the ectoderm. And then also any nerves that are going to be innervating and going into that structure also came from ectoderm. So it's not just the mesoderm that forms it. The mesoderm just happens to form kind of this interior structure going on there. As for like endoderm, there's not really going to be any endoderm going on in the appendicular skeleton. But that is a good question. So, finishing off with that idea of dermatomes and myotomes, on the human body, there are a whole bunch of different nerves and levels coming out of our spinal cord. And those are important for being able to tell pathologies and injuries and localizing where those problems might be going on. The dermatomes are sensory patterns kind of over the skin or the dermis, and that's kind of where you get your sensation from. So if I'm kind of rubbing my hands together and I'm getting that sensation back to my brain, that's traveling through C6, C7, and C8. The C is referring to the cervical vertebra, which are kind of divided up into 3 levels. There's cervical, thoracic, which is like mid-back, and lumbar, which is lower back. And they all have different numbers, as you can see here on the side, but that's a bit out of our scope. But what's important about that is because when someone has numbness or tingling in one of the distributions of a nerve, that will follow a dermatomal pattern. So you can kind of trace that back up and kind of get an idea for either the mechanism of injury or where there might be a problem. The myotome is kind of a functional version of that, whereas dermatome is just I'm rubbing my hands together and I'm feeling it. Myotome is, okay, can I bend my fingers? Can I extend my leg? Are my nerves functioning and reaching the muscles that they're intended to reach? And so something you can think of for a practical application of this is reflexes. Like when you're hitting someone on the knee, the patellar reflex, that's related to L4. If someone has a good reflex, you hit it and their knee kind of twitches out like that, you know that their L4 is likely working well. Whereas if that wasn't working, then you would know there's either a problem at L4 or possibly above it, because the spinal cord is kind of one continuous unit. So if something affects a level above, it's also going to knock everything else out below it, kind of like a power line. But this is not something that is too important for you guys to get lost in the weeds on. This is more for like understanding what the importance is of these topics. All right, so just to finish up, I have a summary here going back over a few of the main, most important things for you guys to know, because I know embryology is a lot. It's a whole new language just about. So some of the main things to take away from this is understanding what somites are, which are derived from paraaxial mesoderm, kind of close to that midline where the spinal cord will eventually be. Those are going to eventually become the dermatomes and myotomes of the limbs and the body. The lateral plate mesoderm is for the appendicular skeleton, which is your arms and legs, anything that is not like central on your core. So not your ribs, not your spinal vertebrae, and those types of things. Those things come from the sclerotomes, which are still located close and coming out from the paraaxial mesoderm in somites. And then another note for those somites is that they start in a head to tail fashion or cranial to caudal, and they're happening alongside the notochord, which is that structure that's really only in the developing embryo. It does have a remnant in adults, which is the nucleus pulposus, but that's kind of like this squishy part of your intervertebral discs in your spinal cord. That's really all that becomes. And then lastly, those somites are going to differentiate into sclerotomes, dermatomes, and myotomes. But those are important for segmentation in the body and the development of different layers and being able to trace those back to midline. And so with that, we've got references and questions, if anybody has any.
We do have a question. What does the intermediate mesoderm develop into?
Okay, that is a good question. So I was actually curious myself when I was putting this all together. The intermediate mesoderm actually will develop into some of your genitourinary system. It splits up and it's got a couple different parts to it. And I'm sure once you get into the embryology of the GI and the urinary system, later on in some of these lectures, that's going to be covered more. But that's what the intermediate mesoderm develops into. If I go back to right here, the intermediate mesoderm, you can almost think that it's kind of in the spot where your kidneys would be, if you imagine in your lower back. So it kind of becomes your ureters and parts of your kidney that are feeding out. All right, any more questions as I skip too far?
And just a reminder to all of you that tune in with us in real time, you can ask any one of the medical students. Doesn't have to be questions about what they presented on. If you want to know more about their journey, how they got to medical school, what was the hardest subject for them, did they take histology before they actually went to medical school? These are all important questions that you could be asking, and I'm sure they'd all be willing to give you answers.
That's very true.
In the meantime, we do have another question. Is the sclerotome a part of the mesoderm?
Okay, that is a good question. So the sclerotome is a subcategory of the mesoderm. So meso, some people like to say meso is muscle, but that's not necessarily true. Mesoderm, it is the muscle, it is your bones, it is also a lot of your connective tissue. So some interesting thing about the mesoderm that we mentioned here. It also becomes parts of your circulatory system. So the blood that's in your body is actually a derivative from the mesoderm. The smooth muscles that are in your arteries as well comes from the mesoderm. So the sclerodome, or yeah, the sclerotome is part of the mesoderm. It comes from that.
So another question is 4-year or 8-year medical school better?
I don't know if I know what an 8-year medical school is. I hope that you would not have to go to medical school for 8 years, that it's just 4.
And I would also encourage Zach and Kyle to chime in here too if you have anything you wanted to add, just so we're not putting Austin on the spot. But
Yeah, I'm not too sure what an 8-year medical school program is. I know there are MD PhD programs that are 8 years, and I know there are BSMD programs where you can get a direct admission to medical. Is that what he's talking about?
yes, yes.
Yeah, so that's that. I mean, if so, I was thinking about applying to BSMD programs as well. And if you know you want to be a doctor right now, 100%. I think it's definitely worth looking into because there are certain BSMD programs that give you obviously direct admission into medical school and make it so you don't have to apply, make it so you don't have to take the MCAT, and you get to bypass all of these just gigantic headaches in your life when it does come time to apply to medical school. But you have to keep in mind, like, you got to be committed to this. Like you could end up your junior year of college falling in love with like PhD or research or business or something completely outside of science. So you got to be really committed to medicine and know that this is what you want to do if you want to go that route.
I'd agree with everything Zach said, and just want to add one more thing in there that you'd also be committing to is the institution when you do that. So I'll just say, I think it is great. You get, normally there's a lot of structure in those programs that offer mentorship throughout your time in there, and they follow up with you, I think, pretty frequently from what I've seen, at least in the programs where I've, interacted with them a little bit. But the other thing I will say is that you are committing to that institution, and if you find that perhaps you'd want to go to a different institution or you find that you want a different experience someplace else, then you still can do those things and apply to those. But I think it's just keeping an open mind is something throughout your time there that I think would be just good to keep in mind.
And maybe another perspective from another faculty member is just the fact that typically those BSMD programs, the day you graduate from high school is the next day you are on your way to your undergrad institution where you're expected to be able to complete the undergrad portion of that program in basically 2 years. That's the 6 years, which means you don't get summers off. You'll never have the opportunity to do a job or to travel. And it means also that you're coming to medical school relatively young. And when you graduate from medical school, you're relatively young. And some of those experiences of having a job or traveling, they allow you to kind of expand your perspective, mature a little bit, because when you graduate from medical school, there are some really serious conversations and serious situations that you will have to discuss with your patients. And sometimes having that maturity and a little bit more time to gain those other perspectives is really important. But there are awesome and excellent BSMD programs out there.
And I think I'll follow up with Dr. Peterson. I think what we're trying to say is that you are still very young. You still have your whole life to look forward to. And I will say I came, med school is one of many amazing STEM programs that you can get involved with. My path was a little different. I have 2 bachelor's, a master's, and a PhD. And that was because I went one route and then I decided to go another route, and I ended up getting a PhD in anatomy. And now I teach medical students. And, sometimes people ask me, oh, so you're not a medical doctor? I was like, no, I make medical doctors. So I teach them, I help create their careers. So there are so many things out there, and that's why we do this competition, is for you to get a sneak peek into what these programs look like, because these lectures are exactly what the med students learn, just a little bit more at your level. When you come to the competition, when you do the virtual competition, it's designed like a written exam you'd take in a health professional kind of environment. And if you do come to the regional competition, those exams are set up like what our medical students take. And I'm so happy here you all are excited, but just take it a day at a time. Your life changes every day, and live in it. And I think, again, that's kind of what we're saying. And we're happy to give all of our point of views and where we're from, and our med students, I know, are more than happy, but I think they would even tell you themselves that it's good to get out there and experience life before you go to med school. I mean, I don't want to speak for them, but I think they would agree.
So we have another question. Would you recommend doing undergrad, then medical school, or the 6-year combined?
I guess I'll chime in. I personally, I would really recommend going to kind of like a regular undergrad. What I did is I would go to college in all my classes in the mornings before 12 p.m. noon. And then from noon until like 7 or 8 p.m., like 4 days a week, that's when I was like coaching gymnastics, or I took a gap year and I was tutoring as well during that time. And being able to have that time where I can do school, but then I can also go and do other things, work a job or hang out with my friends and make a whole bunch of memories. I don't think there's anything that I would trade for the relationships that I got to build during that time, the memories I have, looking back and scrolling through like my photo albums and those types of things. I think that it's really important to be able to develop those relationships, understand like what hobbies you have and what other interests you have outside of school and medicine, so that when you do pursue medicine, yes, like I love it and that's what I'm in like full time right now, but I don't have any regrets about getting to do other things.
Yeah, I also add on, like, if you're like really, really set on going to medical school, then I think maybe a BSMD program might just save some hassle or stress later down the line. But like, if you have like any sort of doubt of like medical school, because I went into like college thinking I wanted to do medicine, but I wasn't like 100% dead sure. And coming from the West Coast, I had no idea that like BSMD programs existed, because there were no like there were none nearby to me. So I just like applied to college like regularly in undergrad. And then I kind of just like explored my options in undergrad, and it's kind of like grew and like just learned from people, met people. And eventually I did kind of I did end up making the decision of medicine. I took one gap year between after graduating college and starting medical school. So to work full time for a year. I think I gained a lot of valuable experience that way. But yeah, I think there's like definitely lots of pros and cons to BSMD versus the traditional pathway. But again, if you're really, really dead set on medicine and you have like good reasons for it, then I think like by all means, a BSMD program is saves the hassle. But I feel like there's a lot of opportunity to like learn about yourself, most importantly, and like figure out like who you are and just life experience in general.
I just want to add one more thing. I I was probably a lot like a lot of you guys. Like I knew I wanted to be a doctor since I was like 10 years old. Okay. And I was similar, like I did, like I got involved in sciencey, medicine-y stuff in high school, and I was all about it. Like medical school is always gonna be there. Like you can go to medical school. Like I have people in my class that are in their 30s with kids. Like it is always gonna be there. You guys should focus right now on like having fun, figuring out what you're interested in, like go live your life a little bit. Like if you guys are seniors in high school, like in less than a year, you're going to be in college. Like focus on making friends, go out, have fun, go to a party, like live your life. Like medicine is always going to be there. Okay.
Yes, we just had a clarification question. So coming from the West, do BSMD programs mean that you're going straight into medical school?
At least here for some of my classmates, because Sydney Kimmel has a linkage program with Penn State. So I know it's technically a 3-year program, but I know a lot of my classmates did like 2 years undergrad and then took a research year or just took a year off before actually starting medical school. But I think it kind of varies from institute to institute.
So another question, would you say that going to med school delays life in any way, like settling down and being stable? Seems like it takes a long time.
Yeah, it does. And it can. It's everyone's different. Like no one's the same way. If you want, like, there are people who have kids in medical school, there are people who have kids in residency. Like it's all about prioritizing what is important to you. If having a family is important to you, then you're going to be able to make time for having a family in medical school. If you just want to grind out and stay single and just, do well in medical school, that's also available. It's just it's what you prioritize.
I would just like to put a theme out here. And I didn't have so much of this at Jefferson last year for when we hosted the regional competition, but I had a few students who were concerned about doing the right classes in high school and doing all this volunteering, and what I'm hearing right now is everybody is coming from different backgrounds. It doesn't sound like anybody did a BSMD program, and they all went in first into undergrad just living their best life and trying to figure out what they wanted to do, and they still got into medical school. Again, you guys are still so young. I didn't have my family. I'm in my 30s and I didn't have my family till now, and I'm doing just fine. I think, again, that's a personal experience. And I 100% agree with Zach. I know, like, you're like, all these people are telling me I'm so young. It's because we are just like you. We've just been here a few more decades, probably. So we've done it. We've been through it. Again, I have 2 bachelor's, a master's, and a PhD. Even though I don't have an MD, I've done the length plus a little bit more of an MD. I think that I want to get to the next question then, because I think it would be a good one for our students. But again, just from my own personal experience, you guys still have time. Take a breath. I know it's important, but I assure you, if this is, especially from my own experience, if it's something you're passionate about, you will do it. It doesn't matter how much you know now, how much experience you get now. If you're passionate about it, if you want to do it, you're going to do it. So I'm going to leave it at that and get to the next question.
Okay, so the next question is, do you have any special memories of patients or shadowing experiences that you think are important to share or that shaped your career? A very probative question.
Yeah, I mean, I guess I can go. I worked so right outside of Ann Arbor, Michigan is a town called Dearborn, Michigan. Um, and Dearborn, Michigan has the largest population of Muslims outside of the Middle East. And it's a very large, like there's a very large immigrant and refugee population that lives there. And I worked in an emergency department in Dearborn, Michigan. So we saw a lot of Muslim patients, a lot of patients who were immigrants and refugees. And I thought it was really interesting to see, like, the cultural considerations that go into taking care of patients who are just like who have certain cultural requirements. Like there were certain patients, like, for example, I remember this woman whose husband was there and he wouldn't allow the doctor to, like, touch her at all for the physical exam. And it was really eye-opening how important it is to take into it. Because like if you have an American patient, you know, you're going to start the physical exam, you're going to touch them, you're going to push on their belly, push on their back, wherever their point of pain is. And I think it's important to understand that like people are from all cultural backgrounds, and you need to take that into account when you're examining a patient, because that could influence how you deliver care to them and how they kind of interpret what you're doing. I think it's really important to keep that in mind.
So another question, is research experience important in entering medical school or excelling later in the career? Would you say it is necessary for people our age to begin now in research programs? How do you really feel, Zach?
Please.
I'll just chime in and say, as when we're interviewing medical students, I'll say at least half that I interview or more don't have experience or started several years after where you guys are now. I'd say that would be way more common than having students that have experience in high school.
And I want to comment too. I know Anato-Bee's goal is to give you money for summer research experiences and summer experiences. Our goal is not to make sure that we want to get you prepared into going into these programs, but the major goal is for you to experience it, for you to want to stay in the program. So I remember one of our students last year went to a summer experience and they didn't like it as much. We interviewed them and they were like, it wasn't for me. And then I was like, that's great, because you went, you tried it, and you're like, I still love science, but this just isn't the program for me. That is our goal here at Anato-Bee, and to help you get through and find your passion and help inspire you to do what you want to do long term. It's not to harp that you need this. You need it to get in. So I wanted to say that too, and I'll let our students take over.
Austin and Kyle, anything to add?
I was going to add my 2 cents. Sorry, Austin and Kyle. So I think a lot of when you're in high school and even in undergrad, you're thinking about, what do I need to do to get where I want to go next, right? What is this box that I need to check? But when I'm, as Dr. Pisciera mentioned, when I'm interviewing candidates for medical school, I don't care if they did research at Harvard with someone who just cured cancer, or if they did research at this tiny little undergraduate institution in Alaska who were discovered like why, you know, squirrels hibernate at certain times, right? The experience itself is the most important thing. Learning how to think critically, learning how to look at science in a different way, right? Instead of just being like, okay, well, I read it and that is the truth and that is what we're moving forward with, right? So being able to look at an article and say, okay, well, this doesn't make sense, and doing that investigation. So it's the skills that you build throughout these different processes. And so Zach was mentioning, right, he had this medical experience before he got to medical school, right? And that helped him, I'm sure is going to help when he goes into 3rd year and 4th year and into residency. And so it's that skill building throughout the experience, and not as much I'm checking the box of all these things that I need to do to get into school.
Yeah, I think to add on to that, I think like if you do anything, you should be doing it with like meaning and purpose. Because I think the number one thing that medical schools are looking for is passion. Because medical school is a very long process, because you have 4 years of you have 4 years of undergrad perhaps, then 4 years of medical school, and then 4 to 8 to 10 years of residency plus fellowship. It's an extremely long journey. And so I mean, the one thing that medical schools don't want is to take someone and then like have them burn out and like drop out of medical school, right? So like I think no matter what you do in terms of experiences, it should be something that you're really passionate about, and then you can like, it's more so like the skills, like was said, like the skills that you can get from it, and like the passion that can be translated from that, and kind of seeing that, I think medical schools will really like just the passion about it. It's also like why medical schools like hobbies. Like they always like talk about hobbies and stuff like that. It's like, what do you do outside of like all this professional grinding that like makes you a person? Like who are you fundamentally, you know? So I think, yeah, again, if you're trying to do something, make sure it's something you want to do, and not just because you're trying to check boxes.
I could not agree more. I think that is the most important thing. And I know speaking like from experience, in undergrad research wasn't like my biggest thing. I don't know if it was just the place that I went didn't have any good opportunities for me or not, but I didn't have very much research at all. But I think what's most important is finding something that kind of suits you, that, like he was saying, you're passionate about and is fitted to your talents. Like, I don't know, me sitting in a lab doing titration and running a bunch of experiments and typing in data and those types of things, that's not the most appealing thing to me. I like being out with people, interacting with them, which is why I did other things, either working in the tutor lab or coaching gymnastics and those types of things in my spare time, because that's what I had the most passion for, and that's what I got a lot of my experience from. And while that's not medical experience, it is experience that is still valuable. It's interacting with people, and I personally got a lot out of it. And I thought that those were ways that I could bring the most benefit to other people. Whereas in research, I don't think that I brought anything that was super, super great to the table compared to those other things.
Right, well, I really appreciate everybody for all of their amazing input. Just want to wrap up this session and say thank you so much again to Kyle, Zach, and Austin for your phenomenal presentations and your just awesome insight for these questions at the end too, as people are navigating some pretty big decisions in life. So we appreciate your insight to that. Thank you again to Doctors Frank, Peterson, Mapes, and Haggerty for our faculty that are helping answering some additional questions as well and giving their 2 cents in there. So we really appreciate all of that and your expertise. And finally, thank you to all of the audience for sticking around so late on Sunday evening and for showing up to our third tutoring session. We are so excited to have you back again in 2 weeks from today on October 27th for our fourth tutoring session. So we will send out the invites to that again just a few days in advance of that, and we're looking forward to it. So I hope everyone has a great night, and we'll see you next time. Bye, everyone.